Okay, welcome to our final week of Living Systems 2. We've got all the lectures wrapped up, including the final lecture, Ecosystems. If you haven't watched the recording yet, make sure you do so. And tonight we're going to focus on some important revision. And of course, this revision is essential going into that final exam. So of course, we can't revise everything, because that would mean that I'd basically have to repeat all the lectures, and that would probably exhaust everyone, including myself. So I'd like to focus on some key points in some of the lectures, starting with harvesting energy. So let's go through to glycolysis. And I want you to consider now, and I need everyone's input here, I want you to consider firstly the biochemical pathway of glycolysis. And can anyone tell me where this occurs in the cell? Does anyone remember? It occurs on the thylakoid membranes. So remember, that's that internal set of membranes found within the chloroplast. Okay, so embedded in the thylakoid membrane within the chloroplast, we have, remember, photosystem 2, which is shown here, and we have photosystem 1, which is shown here. And we also have this protein complex here, which is called the cytochrome BF complex. And then over to the right in purple, we have our ATP synthase protein complex. Very much like the ATP synthase that we just saw in the electron transport chain found in the mitochondrion. All right, so inputs and outputs. First, number one, I want you to tell me what drives this process. Where does the energy come from? Light. Good. Okay, if there's anything that you remember from this lecture, remember light energy. Here it is, that drives the process of photosynthesis. Yep. And who remembers what those little packets of light energy are called? Photons. Correct. Thank you. Okay, so light is obviously a critical input. Tell me what else you can see in terms of inputs or outputs. Water, yes. So here we go. Water is an essential input, and we've got NADP+, yes. Here we go, that's an input. ADP, here we go, there's another input, correct. CO2, save that one for the next part of photosynthesis. That's for the light independent reactions, but good that you brought it up. Okay. Yeah, so everyone's getting the hang of this. What are the two most important outputs from this part of photosynthesis? So what is the primary aim? That's one, yes, is to produce ATP, and there's another one. There's another primary output. So this is one, ATP, yes, correct, and NADPH. So remember these two as being the two most important outputs from the light-dependent reactions of photosynthesis. Correct. Good. Now, we just need to mention something else here. Here we go. Let's go back to our water molecule. Okay, what happens to this water molecule? Why is water required in the process of photosynthesis? What's the requirement for water? Does anyone remember what water is providing? Hydrogen, as in protons, yep. So we can see protons, but electrons. So the electrons that move through this electron transport type of chain here, they have to be replaced. They have to be replaced. And they're actually replaced as a result of the splitting of water molecules. Okay, so remember that a water molecule can be split, not an easy task to split water. That's done by an enzyme component. It's not indicated here, but there's an enzyme component of photosystem 2 that does exactly that. It can split water molecules, yielding protons, electrons, which are used to recharge this reaction centre, the P680 reaction centre. And as a result, an output, a byproduct, a waste product is produced. Who can see it? Who can see that waste product? It's a waste product for the plant, but it's very, very important for us. Oxygen. Excellent. So here's another output, oxygen. And remember, photosystem 2 is the oxygen evolving complex, because that enzyme that splits the water molecule is found in photosystem 2, but not in photosystem 1. And that's why it's widely believed that photosystem 1 was the first photosystem to evolve, and photosystem 2 evolved later on to start producing oxygen and evolving oxygen into the atmosphere. Okay, so have I missed anything? Have we missed anything? No, we've got it all covered. All right, terrific. So remember, light dependent, because of course we need that energy derived from sunlight to actually drive this process. All right, now let's look at the second part of C3 photosynthesis. And it used to be called the dark reactions because it doesn't require light, not because it happens in the dark, it just doesn't require light. But it's now more commonly referred to as the light independent reactions. All right, firstly, where does this part of photosynthesis occur in the cell? In the stroma, correct. So it occurs, yes, it occurs in the mitochondrion, in the chloroplast, but in the stroma of the chloroplast. So it doesn't occur on those thylakoid membranes. It just occurs within that fluid interior of the chloroplast organelle. All right, good. The light-independent reactions of C3 photosynthesis is referred to as the Calvin-Benson cycle, as you can see here. All right, so let's start looking at inputs and outputs, but let's start firstly with a major input. Major input into the light, into the Calvin-Benson cycle. Carbon dioxide, yes, absolutely, here it is. Carbon dioxide. After all, this is the carbon fixation part of photosynthesis. This is how plants get their carbon. Okay, tell me the major and final product of the Calvin-Benson cycle. What is the primary aim? Sucrose. Okay, sucrose. Here it is. So the photosynthesis is geared towards producing carbohydrates. Yeah. So you can say that sucrose, or more specifically, half a sucrose is produced from that turn of the Calvin-Benson cycle. If you wanted to be more specific, you would say that this 3-carbon PGAL molecule, this 3-carbon PGAL molecule is the first carbohydrate that's synthesized as a result of C3 photosynthesis. Yeah. Or you could say that glucose, here we have a 6-carbon glucose or fructose molecule that's being produced. Okay, so anyway, it doesn't matter how you say it, whether you refer to it as a carbohydrate, whether you refer to it as a sugar or a photosynthate. I'll accept any of those. All right, good. P1. Oh, okay, good question. This here is just referring to the release of an inorganic phosphate. That's just the abbreviation. P with that little I, that lowercase I, as a subscript refers to inorganic phosphate. That's all that's referring to. Okay, all right, sorry, can I just pause this for just a minute? I'll just be back in 1 minute. No. You're enjoying this too much. All right, just give me a second. I won't actually pause it. I'll just put myself on mute, actually. Okay, sorry about that. All right, where are we? Inputs and outputs. We've had time to look at this, and we've come up with carbon dioxide and as an important input, and carbohydrates or sugars as important outputs. Okay. And I've got noisy visitors. Okay, does anyone see anything else important? So remember the final products, the major products from the first part of photosynthesis drive the Calvin-Benson cycle. Can anyone see those? Yes, correct. Excellent. Here we go. So here are the major products from the first part of photosynthesis. ATP, NADPH. So they are critical inputs. Remember, Calvin-Benson cycle is very energy intensive. So it requires the input of lots of ATP and lots of NADPH. Okay, outputs here. And we've also got the input of 6 ATP molecules here. Outputs, yes, ADP, NADP plus are also outputs here. Okay, good. So that's photosynthesis. Now let's turn our attention to Lecture 12, Cell Division. All right, so remember that we looked at these two major types of cell division processes, mitosis and meiosis. So let's think now about the differences between the two. Differences between mitosis and meiosis. And there's a couple of differences that you can probably see here. Yes, correct. Mitosis produces cells that are genetically identical to each other and identical to the parent cell. Obviously, that's different in meiosis. Meiosis produces daughter cells that are genetically different to each other and genetically different to the original parent cell. Okay, good. That's one important difference. What else? What about the types of cells that undergo mitosis versus the types of cells that undergo meiosis? So major differences between these two. Perfect. Somatic cells. So somatic cells undergo mitosis, but reproductive cells, remember cells that will give rise to gametes, undergo meiosis. Okay, excellent. That's two differences. What about the number of cells, the number of daughter cells that are produced? Meiosis produces only half. I think I know where you're going with that, but we need to explain it a little bit more clearly. Half the chromosomes, yes. So remember, mitosis will give rise to diploid cells, that is cells that contain 2 copies of each chromosome. Yep, the full complement of chromosomes, basically. Whereas the cells produced by meiosis, they're not diploid, they are haploid. That is, they contain only 1 copy of each chromosome. Good. That's another important difference. Mitosis gives rise to diploid cells, whereas meiosis gives rise to haploid cells. Okay, excellent. And what else? One more. So just going back to what I said earlier, what about the number of cells that are produced? So when a cell divides by So in mitosis, okay, so this cell divides by mitosis. That's right, that cell divides to produce 2 cells, but when a cell divides by meiosis, it will actually produce 4 cells. Okay, good. All right, now, let me clear that. Get rid of my pen. All right, let's revisit the cell cycle and the stages that we find within the cell cycle. All right, so let's think about the major phases, if you like, within the cell cycle. So it starts here with the formation of a new cell, and the first phase is called interphase. That's followed by mitosis, and it ends with cytokinesis. Okay, so they're the major phases of the cell cycle: interphase, mitosis, and cytokinesis. All right, let's look at interphase and break this down. All right, who can tell me the stages within interphase? Just looking at this cell cycle diagram. We can break down interphase into several different stages. What do we call them? Not yet. No, that comes with in mitosis. So interphase occurs G1, correct? Yes. So we've got G1 here. So that's the first part of interphase. S, here it is here. Chromosome duplication phase is the S phase or synthesis phase. And then G2 here. And there's one more possible stage within G0. There it is there. Excellent. Okay, so 4 components or 4 separate stages within interphase before the cell enters mitosis. All right, good. Let's now move on to mitosis. Okay, so the cell has proceeded through interphase, then it enters the first part of mitosis. And the first part's called prophase. The second part of mitosis is prometaphase. The third part is metaphase, then anaphase, and the final part of mitosis is telophase. So let's go back. And who has some way of remembering those different parts of mitosis? I can share with you what students have come up with. And they have come up with, well, at least one student has. They just remember PPMAT. I don't know if it means anything, but I think it's easy to remember. PPMAT. Prophase, prometaphase, metaphase, anaphase, telophase. All right, have a look at the first part of mitosis, prophase, and let's look at the major processes that occur, the major events, I should say, that occur during prophase. Okay, who can list them briefly for me? The major events, which they're all described here, but I just need you to pick out these major key points. So during prophase. So we can start to see something here. Yes, chromatids condense. Yeah, here we go. Sister chromatids condense. In other words, the DNA now becomes visible using just a standard compound light microscope. Okay, something else, spindle starts to form. Here it is. You can see it in green, or we can see it here. The mitotic spindle starts to form. Okay, they're the 2 major events in prophase. Let's move on to prometaphase, second part of mitosis. What are the important events that occur here? Something's broken down completely in prometaphase. That's right. The nuclear membrane has completely broken down. So you can see it here. Oops, so you can see the nuclear membrane here. Here, it's completely gone. Yep, nuclear membrane has completely broken down. Something else, what's completely formed now in prometaphase? The spindle, the mitotic spindle, yep, is fully formed here. And correct, the sister chromatids are now attached to the fibres of the mitotic spindle. Good. Let's go to the next part of mitosis, metaphase. Okay, one major event, important event that occurs during metaphase. It's a pretty easy one, metaphase, I think, and anaphase too. Chromatids align, absolutely. So here they are, sister chromatids, they align, and where do they align? They're aligning along something, a region that's referred to as something. They're aligning along the metaphase plate, correct. Yep, so this middle region here of the mitotic spindle. All right, next part of mitosis, anaphase, the most dramatic part of mitosis. And what's the major event here in anaphase? Chromatids separate. Absolutely correct. So the sister chromatids separate from each other. The chromatids separate from each other, and they're now referred to as chromosomes, and they move to each pole. They move to the poles, exactly right. Okay, final stage. Sorry, I'm getting distracted here. Final stage of mitosis, telophase. What occurs during telophase? Telophase. Membranes reform. Correct. Correct. So the nuclear membranes start to reform. One there and one here. Now, cleavage, that's a very good point. Cleavage, you can see the formation of the cleavage furrow. This is already occurring even before we get to telophase. But the actual cleavage event itself is not considered to be part of mitosis. The cleavage event of the cell into two cells is considered to be a separate stage called cytokinesis. So just keep that one in mind. But after mitosis, then you have the completion of cytokinesis, and that's considered a separate stage. All right, so important event, membranes, nuclear membranes reform, and you could say that the cleavage furrows appear, and you could also say that the chromosomes start to decondense so that they'll no longer be visible. All right, terrific. Let's move on to Lecture 13, Inheritance. Okay, now on the final exam, I think I've mentioned this before, you'll be required to perform a genetic cross. So this particular slide is is critical in terms of understanding how the cross proceeds and also how to work out the ratios and proportions of offspring. So remember we talked about crossing this pure breeding parent line, which is pure breeding for yellow seeds. And remember we can say true breeding or pure breeding. Crossing that to parent line number 2, which is pure breeding for green seeds. As a result of this genetic cross, the F1 offspring, so the F1 generation, which is the offspring, are produced, and then 2 individuals from the F1 generation are crossed. That's referred to as a monohybrid cross because they differ just in one trait, hence monohybrid. So 2 of these individuals are crossed to produce an F2 generation, and the F2 generation are represented here within a Punnett square. Okay, so that's just an overview. I'd like to actually work through an example. And to do that, I'm going to need my whiteboard, which I'll just bring that up. There we go. All right, and I'll use the example that we went through today. We're looking at coat colour in horses, and we have a horse that is big B, so dominant alleles there, which represents black coat colour. And that horse is going to be mated with, or crossed with, a little b, and little b is the recessive alleles, which represents white coat colour. I should write that down. So we've got black coat colour. So it's the dominant allele, black and white. And this is the parental generation. So we're going to perform this genetic cross. All right, let's just allocate, just for the purpose of this exercise, let's allocate male and female. So we'll say that this black horse is the male, and this is the symbol for male, and let's say the white horse is the female, and the symbol for a female. Okay, so let's just revise a few terms. Who can tell me what the genotype is of the male horse, of the stallion? What is his genotype for coat colour? Yeah, it is dominant. Yeah. What is the specific genotype though? Tell me what the genotype is. It's probably too simple a question. This is the genotype written here. So his genotype is big B, big B. And you're right, it's a dominant phenotype, and there's a little bit more to it. So you can say that this genotype is dominant what? Heterozygous? Is he heterozygous at this particular gene locus? Or is he homozygous for the coat colour gene? Does anyone remember that? Homozygous versus heterozygous. Homozygous is correct. So remember when he's homozygous, yeah. When 2 alleles, which are shown here, 2 alleles for the one gene and they're identical to each other, that's referred to as homozygous. So both of these alleles are the dominant alleles. So we say that his genotype of big B, big B for the coat colour gene is dominant homozygous. Correct. All right. Now tell me what the type is for the mare, the female. Her genotype is obviously little b, little b. So how do you describe that? How do you describe this? In the same way that we described the male. Correct. Good. Recessive homozygous. So these are both recessive alleles, but they're exactly the same as each other, so we still say homozygous. Okay, good. Now, we're going to move on to, so we've got, sorry, so that was the P or parental generation. Now we're going to look at the F1. So when you're asked to perform a genetic cross, of course you go to the F1 generation. And I'm just going to draw something here. Okay, so what's this called here? What's that referred to? Punnett square. Good. All right, that's a Punnett square. And before you start filling in the Punnett square, you need to put something here on the outside. What do we put on the outside of a Punnett square? Remember, it's the offspring. So the F1 generation offspring will be incorporated on the inside of the Punnett square. On the outside, we're putting, yes, yep, you're right, you're both correct, but I want to know what it is. What is it referring to? What are these letters that we put on the outside actually referring to? What do they mean? What do they represent? Alleles, yes, they do represent alleles, yep. And those alleles that are written on the outside here are found in what types of cells? So remember, yes, male and female what? Male and female gametes. Perfect, excellent. All right, so here, on the outside here, we actually first start with the male. So there's the male, and of course what we're looking at here is the diploid state, because there's two alleles for this particular gene. So we must be looking at a pair of chromosomes. Yeah. And remember that gametes are not diploid, they're haploid. So here's the male gamete, so the sperm. So the sperm can carry what genotype? Haploid gamete will carry what genotype? Produce this stallion here. Okay, a couple of you mentioned something earlier. What gamete, what would be the genotype of the sperm, the haploid sperm produced by this male horse? Okay, the sperm will carry this allele, or it will carry this allele, one or the other. In other words, all the sperm cells will carry the same big B allele. So we don't even need to have that one there. But I put it there just to illustrate a concept. Let's look at the female gametes. And I might put them down the bottom here because I'm running out of room. So the female gametes, and I'll put them along here. Normally you put them at the top, but it doesn't really matter. Let's put them here. What would be the haploid genotype of the female egg cells? So that's what she would have in her diploid cells. But remember, when some of those diploid cells divide by meiosis to produce egg cells, they will only contain one copy or the other. Little b. So little b, so the egg cells will contain this allele as the haploid genotype, or this allele here. So in other words, all the egg cells will contain the little b allele. Yeah. All right, now we can start combining these gametes, of course, in the process of fertilisation to produce the diploid zygote. How do we do that? Big B sperm fertilises a little b egg to produce this zygote. In other words, this will be a possibility for an individual offspring in the F1 generation. We can go big B, little b, big B sperm, little b egg cell, same here, big B sperm fertilises little b egg cell to produce this diploid zygote, and the same thing here. Okay, so what proportion of the offspring as a result of this genetic cross will be heterozygous? Obviously 100%, all of them. 100% of the F1 generation will be heterozygotes. And remember, they're heterozygotes because the two alleles for this particular gene, for the coat colour gene, are different from each other. Okay, moving on. Now we're going to perform a monohybrid cross, which means that we're going to take a big B, little b from the F1 generation and cross that to another heterozygote from that generation, from the F1 generation, to produce offspring in our F2. So we draw our Punnett square and we consider firstly those haploid gametes. Okay, Andrew, you're too fast. Too fast for me. Or am I just going too slow? All right, so let's look at placing our haploid gametes on the outside of this Punnett square. And just to make it a little bit easier, let's just allocate male and female, which you don't have to do, but I think every little bit helps here, just to try and visualise what's going on. All right, so there's our female, there's our male. So let's start with our male gametes. There's the male symbol. So what are the possible haploid gametes that can be produced from this F2 individual? Well, either big B or little b sperm. So let's pop them over here. So the sperm may be big B and they may be little b. Okay, what about the haploid eggs produced by this female mare, this horse. Same thing applies. Egg cells may have the genotype big B, or they might inherit the other allele, little b. Okay, so what are the possible genotypes for the offspring? Big B sperm, big B egg, fertilisation will give rise to an F2 individual with the genotype big B, big B. Okay, let's try this one. Big B, little b. What about down here? So we've got little B sperm fertilising a big B egg cell. Okay, I'm actually writing that the wrong way around, but it doesn't matter. And then here we have a little b egg cell being fertilised by a little b sperm to produce an individual which has a little b phenotype. Okay, so they're the possibilities for the F2 generation. In other words, every individual that's produced as a result of this cross, this monohybrid cross here, has a 1 in 4 chance, or as Andrews put it, 25% chance of being this genotype. Every individual, every offspring that's produced has a 1, 2 in 4 chance, or 50% chance of having a big B, little b genotype. Every offspring that is born has a 1 in 4 chance of being little b. So that's how it works. So let's now consider and just revise what's meant by genotypic ratio. Genotypic ratio. Who remembers what this ratio is? Genotypic ratio, as a result of a monohybrid cross where we have dominant and recessive. Correct. 1, 2, 1. Good. 1 to 1 ratio. What does this 1 mean? What's that referring to? This number 1, that's referring to big B. And what do we call this again? This big B. Correct, homozygous dominant. What's this number 2 referring to? Yep, correct, good. It's referring to big B, little b, which we refer to as heterozygotes. So remember this, you need to describe this. And the last one in this ratio is the which genotype? Okay, little b. Yes, little b, this one here. And we refer to that as homozygous recessive. Correct. Good. All right, now one more ratio. So that's the genotypic ratio. What about the phenotypic ratio? Who remembers this one? What is the ratio? Phenotypic. So remember phenotype, what you can actually see. In this case, we'll be talking about black and white coat colour. Ratio of the observed phenotypes. 3 to 1, correct. There we go. Amelia, are you back from overseas or are you joining us from overseas? Oh, yes, you're back. Welcome back. Okay, phenotypic and yeah, I can imagine. All right, phenotypic ratio, 3 to 1. What is the 3 referring to? 3 what? Okay, 3 what? 3 black, yes. 3 black. Yeah, which ones are the black ones? The black foals. Which ones are black? Correct, good. This one's black, this one's black, and this one. And of course, this number, this part of the ratio refers to the white offspring, and of course this one would be white. Little b, little b. All right, fantastic. Let us now go back to our lectures. Okay, lecture 13. Got here, lecture 15. Right, does anyone need a break? Soon. All right, tell me when you need a break. I'm sure you will after this part here. Okay, let us revise our technique of PCR, that is the polymerase chain reaction. So remember, this is a technique, very widely used technique, that enables you to isolate and amplify a specific region of DNA. So you could target any gene of interest within the genome, within a collection of DNA fragments that you've extracted from your grapevine or from your horse or from your own blood cells, basically from any living organism or from a culture of bacteria or a culture of yeast. Yeah, you've extracted all this DNA, but you're really interested in one particular gene from that genome. PCR is the way to go. So we're going to revise this technique, okay, which is shown here and also shown on this slide here. Okay. Sometimes okay, is everyone still there? I think I dropped out there. I dropped out, didn't I? But no one else did. That's interesting. Okay, all right, I'm back. All right, we're going to look at PCR, but as I said, I would like to draw it out from scratch. It's just a different way of looking at it, instead of always looking at those lecture slides. All right, polymerase chain reaction. Let's start by thinking about the requirements for this particular reaction. What goes in to the polymerase chain reaction? Okay, so just get that happening. Requirements. So obviously, as I was saying before, you would need DNA. DNA that's been extracted from cells or tissues from plants or an animal or yeast or bacteria, and we call that DNA template DNA because it's going to serve as a template to amplify your gene of interest. So let's get that down. Template DNA. Okay, what else do you think we need to perform the polymerase chain reaction? And if you can't remember any of it, that's fine. After all, this is revision. Okay, what else do you need for the polymerase? Primers, very good. Primers, otherwise known as oligos, but primers is perfectly fine. We need primers. Don't worry if you don't remember what they are. We're just thinking about what goes into it for now. And then we'll come back to that. Something else. So we've got a template DNA. We have our primers. Do we need heat? Yes, we do need heat. I'll just put that over there. Because we certainly need to heat up the reaction. Template DNA primers. What about something that catalyses the reaction? What would you have that catalyses a reaction? Something that will copy the DNA. Enzyme, yes. We need an enzyme. And obviously that enzyme has to withstand high temperatures. So the enzyme has to be thermostable. Does anyone remember where these enzymes come from? Thermostable enzyme. No, if you don't remember, that's fine. Thermostable enzyme. No. Okay, the first enzyme that was used in the PCR reaction was actually isolated from a bacteria that was isolated from hot springs. So, of course, if you have bacteria that live in these really hot springs, they must have thermostable enzymes that don't denature when they're heated up. All right, so we've got template DNA, we've got primers, we've got enzymes, and remember when an enzyme is copying DNA, then it needs the building blocks of DNA. It needs to incorporate the building blocks of DNA. And what are the building blocks of DNA? What are they called? What are the basic building blocks of DNA? And there's 4 of them. There's a clue. 4 different building blocks of DNA. Anyone remember those? One's called A. One's called, that's them. Yes. A, adenine, thymine, correct, guanine, cytosine. So they're the 4 . All right, so you need to add them. If you don't add them to the reaction, then the enzyme doesn't have those building blocks required to copy your gene of interest. All right, terrific. So now let's look at the process. Clear that. And let's start with step 1. Step 1. Step 1, it's performed at a fairly high temperature, approximately 95 degrees Celsius. And it's called DNA denaturation. Denaturation. DNA denaturation. So what's happening during this first step? What happens to your DNA when you heat it up to 95 degrees Celsius? So imagine you've got your double-stranded DNA, because that's how DNA exists. No, it doesn't die. That's very good, but no, not quite. It doesn't die. It doesn't actually destroy. Actually, it will destroy it if you heated that DNA for, say, I don't know what the minimum would be, but I can tell you definitely if you keep heating that DNA at that temperature for 2 hours, yes, you will destroy it. You will destroy it, definitely. And I can tell you that because I've done it accidentally. All right, it unravels, it splits, it becomes single-stranded, all correct. So what happens is that you're separating those 2 strands from each other. So your double-stranded DNA becomes 2 single strands of DNA. That's what's referred to as DNA denaturation, splitting those 2 strands apart, and we now have those individual strands. So how does heating actually achieve that? What is it breaking? What is that heating actually breaking to enable the denaturation of the DNA to occur? Hydrogen bonds, correct. So normally, remember, there's all these hydrogen bonds between the nucleotides of these 2 strands, and that's what holds the 2 strands of DNA together. Heat will disrupt that hydrogen bonding and allow the 2 strands to split apart. Terrific. All right, let's move on to, just clear that, and let's move on to step 2. Step 2, the reaction, the PCR reaction, is cooled down to approximately 55 degrees Celsius. And this is a temperature that the operator, if you like, has to change depending on certain parameters, but don't worry about that. Okay, this part of the PCR reaction we can call primer annealing. Primer annealing step. So when it's cooled down, when the reaction is cooled down to approximately this temperature, it's not cool enough to allow the 2 strands of DNA to come together again, but it is cool enough to allow these very, very short primers to bind to each of those single strands of DNA. I'll show you what that looks like. So let's say we have 1 strand there and we have another strand there. And let's say that this is the gene, this is the region that you're interested in there. And of course, that's part of it on the opposite strand there. And then in the mix, you have little primers. So one will bind to one side of the gene and the other one will bind or anneal. That's what we mean by anneal. These little primers will bind or anneal. So they're basically added to the mix and they're all floating around, these little primers. And the reason that they bind to where they're binding is because they've been designed to be complementary to those regions of the DNA. In other words, you have to know the nucleotide sequence here to make this primer that's complementary to that sequence, and you need to know the nucleotide sequence in this region of the DNA so that you can design and synthesize this little primer. All right, so that's primer annealing. And who remembers the importance of this primer annealing? What do the primers actually provide, enable for this reaction to proceed? They're providing something. Does anyone remember that? I should have drawn them a bit bigger. Not because they're big, but because I won't be able to show you what they're providing. They enable something. Yeah, they do, yep. They enable something. They are essential to enable something to do with the enzyme, to enable the enzyme to make copies. But do you remember why? What is it that they're providing so that the enzyme can actually come in? No, not quite. They're providing something here on this end and on this end. A hydroxyl group. They're actually providing this OH group. Yeah. You might not recall, but the enzyme that copies DNA, called DNA polymerase, can't start incorporating the nucleotides required to synthesize a copy of this gene unless it has this freely available hydroxyl group, this OH group. Only then can the enzyme come in and start incorporating these nucleotides in this other copy. Okay, so that's primer annealing. Now let's go to step 3. Where are we? Okay, so the primers have annealed. Step 4, temperature goes up to approximately 72 degrees Celsius. And this particular step of the PCR reaction, sorry, did I say step 4? I should say step 3. That's 72 degrees. And what happens at this temperature is that it actually provides the optimum for enzyme activity to occur. So this is where the DNA polymerase enzyme that you've added to the reaction can start extending the DNA that is copying each strand of DNA. So we call this DNA extension. But think of it as copying your gene of interest. DNA extension. Okay, once again, our 2 strands of DNA and our gene of interest. This is our DNA of interest that we're trying to amplify. We're trying to amplify it because we want to study that particular gene. We might want to study the function of that gene. Okay, we have a primer here. And I'll just draw it as a very big primer. And we have a primer here. And on this end, we have a hydroxyl group. And on this end, we have a hydroxyl. And during this DNA extension step, the enzyme can come in. So here's our enzyme, and it comes in here and it starts to copy the DNA in this direction. So it's copying your gene of interest. And here's another DNA polymerase enzyme, and that comes in and starts copying the other strand of DNA in this direction. Okay. So at the end of this first round of the polymerase chain reaction, remember we started with 2 strands of DNA. At the end, we would have 1, 2, 3, 4. So we've gone from 2 strands of DNA to 4 strands of DNA. So we've doubled the amount of DNA. And of course, this cycle is repeated over and over again until you theoretically could have 1 trillion copies of your gene of interest. And that's how the polymerase chain reaction works. All right, any questions for me on this part? No? Everyone happy with that? Now do you need a break? All okay? No? All good. All right, does anyone need a break before we move on? 13, short one. All right, you want 5 minutes? Okay, 8:05. All right, let's come back at 8:10. See you soon. Okay. I dropped out again. Was that just me or everyone else dropped out as well? Really, just me? Yeah. Oh, my Wi-Fi just has to hold on a little bit longer. All right. Let's see what we've got here. Lecture 15. Okay, we're nearly there. Nearly there. All right, who remembers our Hardy-Weinberg hypothetical population? The Hardy-Weinberg model, this hypothetical ideal population that reaches an equilibrium and does not evolve. And this is based on certain assumptions that are listed here. So remember, individuals mate at random, population is so large it's not affected by genetic drift, there's no mutation occurring, no migration, and no natural selection. So these are the assumptions. And remember that any deviations from these assumptions, that is, if one or more of these assumptions is not met, then allele frequencies may change and the population is said to evolve. So let's just look at one example of a deviation from a Hardy-Weinberg assumption. So here's one of the assumptions, and that is that individuals mate at random. So this is what would occur in this hypothetical population. Mating would be completely at random. But of course, that's not what happens in naturally occurring populations. What does occur is assortative mating, where a trait has an effect on mate choice. So this is the deviation from the Hardy-Weinberg assumption. And you could say that assortative mating can occur in two different ways. So you may observe positive assortative mating, where mates will select, a mate will select another mate based on similar phenotype, so similar eye colour or similar height, for example, in humans. Or you can mention negative assortative mating, so another type of assortative mating. And this is where mate choice is based on different phenotypes. And we use this example of mate choice based on this MHC locus. That is that genetic region that contains lots of genes involved in the immune response. All right, so that's an example of Hardy-Weinberg assumption and a deviation, deviations from that assumption, which would occur in natural populations. All right, and I won't go through the rest. That was just to give you an example of what we mean by Hardy-Weinberg assumptions and the deviations. All right, time to look at the final exam. Are you all ready? Yes? Maybe? Maybe? Okay. All right. Let's start with the date. So, of course, it's the first, the exam will occur on the first day of the exam period, Monday the 30th of October. We're currently in week 13 of semester, so this is the last week of semester. Next week, you should all have the week off, and then the following week is the beginning of the exam period. And apparently most subjects don't have exams anymore, which I think would make students very happy. So is it true that most of you, or maybe all of you, only have one exam? Only this one. What about everyone else? Yep, you too, Andrew. Okay, some of you have got 2 exams. So you've got an exam. Okay, what's your other exam for? What subject? What other subject has the exam? Wine of that. Okay. All right, so you're doing very different subjects. Vineyard management has an exam, international wine styles, wine evaluation. Okay. And that oral exam is for what subject, Daniel? Is that wine evaluation? Okay. All right, vineyard management. All right. Okay, 2 exams, not bad. Certainly beats the days where you had an exam for every subject. All right, time, 9:30 is the start. It's a very intense exam, just like the final exam for Living Systems 1. So the key is obviously preparation. If you're really super well prepared for the exam, then you will be able to complete it and do exceptionally well. So as I said to the students this morning, every year there are a small number of students that seem to be exceptionally well prepared, and they will come out and not only complete every question, but they'll achieve, a final score of over 90% for the final exam. So I think that just shows that it can be done, but it just depends obviously on the amount of work that you want to put into the preparation. All right, the weighting for this final exam is 30%. It's at Epping, of course, in Building B. So that building next to the cafeteria, in room 127, 128. Don't forget to bring your student card. This is important because there will be external invigilators supervising the exam. They don't know you guys, so they need to have photo evidence of your identity. All right, let's look at exam structure. Section A. All right, that's not me. Andrew, what do you mean by that's not me? That's not you that will get over 90%? Is that what you mean? But it's possible. It's more than possible. All right. Doesn't recall. It takes a lot of revision, a lot of time. To get marks like that in any exam, it means you're putting in many hours of revision, rote learning, revising. Yeah, it's certainly not an easy task. All right, section A of the exam is multiple choice. Each question is worth 1 mark and there are 49 questions. Okay, remember during reading time, don't waste it. Of course, you read through or rather skim through the exam, get your head around the structure, the types of questions, and then use the rest of that reading time to start answering multiple choice questions in your mind. Remember, don't write anything, don't mark the answers with your fingernail or anything like that. But you can certainly start answering the first 10 to 20 questions within reading time. All right, section B, true or false questions. Each question is worth 1 mark and there are 20 questions in this section. Section C, short answer questions and some long answer questions. Each question is worth 2, 4 or 5 marks each for a total of 36 marks, and there are 8 questions in section C. Keep in mind that some of those questions will ask you to select and answer, for example, 2 of the following 4 questions. So within one question, you may be answering 2 different questions. But at least you get that choice, a bit of a choice there. Okay, make sure you revise all your lecture material. Final exam will cover lectures from lecture 11 through to lecture 23, our final lecture. In other words, from harvesting energy, which is lecture 11, through to our final ecosystems lecture. Okay, 11 to 23. Okay, tutorial questions. I don't need to remind you how important these are, particularly for those multiple choice and true and false questions. Make sure you understand cell division, which we've covered tonight, genetic crosses, including that monohybrid cross, glycolysis, citric acid cycle, electron transport chain, and C3 photosynthesis, which we've also covered. Polymerase chain reaction, we've just looked at. Revise your gametogenesis, that is the formation of gametes in plants and formation of gametes in animals. Okay, revise what's meant by evolution, natural selection, genetic drift, bottlenecks, founder effect, and have some idea about some of the Hardy-Weinberg assumptions. So we just looked at one example. Hardy-Weinberg assumptions and deviations from these assumptions in a natural population. Okay, our final one, biogeochemical cycles. So if you haven't watched that final lecture on ecosystems yet, you'll see that there are descriptions of some of the nutrient cycles, which are referred to as biogeochemical cycles. So for example, we talk about the nitrogen cycle, phosphorus cycle, carbon cycle, and water cycle. So make sure that you're familiar with, say, 2 of those cycles, 2 of those nutrient cycles. And that is it. That concludes living systems. Yay. You got through it. Thank you, everyone. It's been fantastic. Fantastic. I've really enjoyed it. Look forward to seeing you all in a couple of weeks. Well done on getting through. Well done. Just 10 weeks. Thank you so much, everyone. It's been really, really tremendous. It's been really great. So thank you to everyone. Yeah, we're not finished yet, but we're nearly there, nearly there. Just got a couple of prac reports for some of you to get in this Friday. And of course, just that final exam. But it's been fantastic. Please, if you remember, remember to remind me to get a photo, a group photo, before you guys all go into the exam. I've always wanted a group photo of the students and never managed to do it. And I think that'll be the perfect opportunity. Yes, this lecture will be up on the system tomorrow, sometime tomorrow. It'll be available, but I'll let you know when I get it up there. All right, you're welcome. All right, have a great 2 weeks. And I'll see you all on exam day. Okay, good night, everyone. Thank you.